We are recording. Thank you everyone for joining us today for the Frontiers in Child Health Research Seminar. We hold the seminar on the second and fourth Mondays from September through June. The series includes speakers with work focused on child health from both UCSF as well as guest speakers. Please let me know via email if you're interested in speaking or would like to suggest a speaker. Looking ahead to our next seminar on March 11th, we'll be welcoming Dr. Mark Peterson from the Division of Neonatology. We're still virtual and using Zoom, and we'll ask everyone to stay muted unless you have a question, and then we'll also open up at the end for questions. Today, we're welcoming Dr. Andrea Garber, Professor of Pediatrics in the Division of Adolescent and Young Adult Medicine. Andrea is the Director of Nutrition for the UCSF Eating Disorders Program. Her research focuses on eating disorders with specific focus on nutritional rehabilitation and body composition, and is funded by the NIH. The title of her talk today is Advances in Assessment and Treatment of Malnutrition Secondary to Eating Disorders. Welcome, Andrea.
Thank you very much. I'm happy to be here, and I will now attempt to share my slides. Good, looked like it worked. Huh, Roberta? Okay, great. Okay, well, thank you, everyone. It's a fun to be able to talk to this group. And just to remind everyone where my work is housed in the eating disorder program at UCSF, here is our fancy, I believe this is our Facebook page, of course, featuring the wrong bridge, but you know, we have to look good online. But the eating disorder program, for those of you who are not familiar with it, our program spans pediatrics and psychiatry. Sarah Buckaloo is our medical director on the pediatric side, and I know you all know Sarah. We have outpatient clinics every single day, including 2 satellite clinics, one in Green Bay, one in Walnut Creek. And we have an inpatient service, usually about 8 kids at a time in a hospital for malnutrition secondary to restrictive eating disorders. We were at least double that during COVID. And in fact, the numbers have never really come down. If anyone has seen any of the studies or reports coming out showing that the rise in eating disorders during COVID is really kind of part of the mental health tsunami that grew during that time. We do also have an intensive family treatment program that was paused during COVID and is just getting started back up, but a week-long intense educational experience for families. And we see about 400 patients per year. So that is where the patients that participate in my clinical trials come from. And I also do provide clinical care directly for patients. So for today, here's what I'd like to cover. Just a little bit of background on anorexia nervosa and malnutrition for those of you who are not operating in this world. I'd like to talk about refeeding or short-term nutritional rehabilitation, the importance of that in the treatment pathway, and then tell you a little bit about our clinical translational work, the STRONG trial that we completed in 2020, the trial that we're doing now, and then just mention a couple of fun new directions we are taking the work. So for the background, I see Naomi Bardock is on the call. I always like to start with background by quoting her very nice paper. Naomi, thank you for this contribution from 2014, an oldie but a goodie, showing that eating disorders are among the top 6 most expensive primary mental health diagnoses in pediatrics. And that really nice work showed that part of the reason eating disorders are so expensive is because of the long and intensive hospitalizations. And even after the first long and intensive hospitalization, almost half of the kids that we provide care for are rehospitalized within the first 12 months. Only about 1/3 of our patients meet full clinical remission criteria at 1 year. And so this is an illness that, despite great advances, there's a long way to go. And unfortunately, eating disorders do remain, or anorexia nervosa is the most deadly psychiatric diagnosis. So that's the bad news. The good news is that there is a lot of intensifying focus around malnutrition. And so all of you in pediatrics are familiar with this new focus with data emerging that about 1 in 3 hospitalized patients are malnourished, and that markers of malnutrition in up to 65% of patients worsen while they're in the hospital. You may also be familiar with the new NIH Strategic Plan for Nutrition Research. There are specific objectives around reducing the burden of malnutrition in hospitalized patients. So our work really squarely fits with those objectives. And hopefully, the NIH will carve out some more money for work in that area. So I always describe patients with anorexia nervosa as a unique population with malnutrition, because the malnutrition in anorexia nervosa is uncomplicated, meaning that these are not patients with critical illness. These are not patients with infection. And so in some ways, this is a model of malnutrition that is somewhat easier to study. And I always, try to simplify things when we're working with patients themselves and trying to describe what we're looking at clinically. We use kind of this state of hibernation to describe why they're coming into the clinic with a low heart rate, low blood pressure, feeling cold. But as you all know, malnutrition is actually not simple. During refeeding, we're trying to reverse some very, very complicated adaptations to starvation. So our patients have appetite dysregulation that contributes to their difficulty renourishing. They arrive in a hypometabolic state. So we do measure they have low resting energy expenditure, but we also take blood samples, and you can see they have low leptin, low T3 when they're admitted. Patients have HPG axis suppression, so the sex hormones are all low. Our new study, we have about 5 boys only, so we're looking at low testosterone in boys, but obviously also low estradiol, LH, and FSH in our female patients. And just in general, when they're admitted to hospital with us, they're in a catabolic state. So they're breaking down protein, they're breaking down their glucose stores, glycogen stores, and they're breaking down fat stores. And we'll talk a little bit about that, but just to impress upon you, or remind you that, although we call this uncomplicated malnutrition, we're really trying to reverse a lot of complicated processes that have become entrenched during the starvation period. But we do stick to the basics. So we really use vital signs very heavily. We have vital sign criteria for admission to our hospital unit. They're shown here. It's low heart rate, low systolic blood pressure, and then orthostatic or positional changes in systolic blood pressure and pulse, low temperature, and then some patients are admitted just for flat out low body weight. And then we use these as nutrition sensitive outcomes. So our main outcomes, and I'll show you for our clinical trials, our main outcome has been the number of days that it takes to reverse these vital sign instabilities. And you'll show you in a minute what that looks like. But first, I want to describe what do we do. So I just said we have our hibernating bear type of patient who is admitted to the hospital for malnutrition, secondary to a restrictive eating disorder, and we start a refeeding protocol. A lot of people really hate the word refeeding because it sounds so forced. Some people call it short-term nutritional rehabilitation, but nutritional rehabilitation really is a very, very long road, and the time in hospital is very short. So I'll show you what we can accomplish. It's something, but it's not much. So what does refeeding look like? It does not look like this. There's no double bacon cheeseburger, fries, and milkshake the day that kids arrive. It looks like a regular old hospital tray. We do all meal-based refeeding, and that's very common in the US. Internationally, there are a few countries. Australia is notable for doing tube-based feeding. So they place a nasogastric tube and they start overnight feeds on all of their patients. It's very uncommon in the US. Most programs use meal-based refeeding. And there you can see we have 6 trays per day. It's not fancy. It's regular. We have very good hospital food at UCSF, but still it's meal that conforms to the dietary guidelines for Americans. So it's somewhere around 30 to 40% fat. We have meal monitors or room sitters. So patients are supervised to ensure that the meals are completed. If they're not completed, they drink a liquid supplement like a Boost. And the supplementation protocol is also not very complicated. They take an adult multivitamin with some extra calcium twice per day. We do replete electrolytes though, and I'll tell you a little bit about that in just a minute. So for those of you who are physicians, of course, you're very familiar with the refeeding syndrome, but that's why I have, the quadruple hamburger with the big arrow line through it, because the biggest concern or the most dire medical consequences during refeeding is the actual development of the refeeding syndrome. So as I said, patients are in this catabolic state of breakdown. They're breaking down glycogen stores and protein. And when you reintroduce nutrients and you get a nice big insulin surge, the cells take up glucose and the electrolytes move from the extracellular to the intracellular space. And you can see really rapid drops, particularly in phosphorus, which is not just being moved from the extracellular to the intracellular space, but it's also being used to make ATP as you turn those metabolic pathways back on. So the physicians look at labs every single day to keep an eye for low phos, low mag, hypomagnesemia, and hypokalemia. And then those electrolytes are repleted as needed. And I'll show you our protocol. But this syndrome actually has been very well known for quite a long time. Some of you may be familiar with the Minnesota starvation experiment. This was done around the time of World War II. It actually was intended to look at refeeding protocols in case those were needed in war-torn regions. And during this study, they did document almost all of the features of the refeeding syndrome. Also, deaths were documented in World War II prisoners at 1951. Since then, the refeeding syndrome, including all of the clinical signs and symptoms, cardiac arrhythmias, cardiac failure, cardiac arrest, delirium, seizures, coma, unfortunately, those have all been documented in patients with anorexia nervosa undergoing refeeding. So as you can imagine, since those documented cases of the refeeding syndrome, the early protocols for refeeding were extremely, extremely cautious. The written protocols are around 1,200 calories per day starting. In Europe, the NICE guidelines, so the kind of the British NIH, so to speak, has had guidelines starting as low as 500 calories per day. So you can imagine, bringing a young person into the hospital to treat their malnutrition and then basically starting them on a starvation diet. And the studies from that time showed that to reverse those vital sign instabilities that I showed took up to 3 weeks of sitting in the hospital. And in fact, when I started doing this work in 2002, Roberta, I think you were here back then. That's when the PCRC was over at Moffitt, and we admitted our patients over there on the 6th floor. And you can see here the data from one of our first studies we published in 2012 on low-calorie refeeding. And what you can see here is that patients, first of all, they were in the hospital on average 18 days. The first patient that we ever enrolled in a clinical trial was an otherwise healthy young person with malnutrition secondary to anorexia nervosa. And she was in the hospital with us for 35 days, which would just be unheard of now. But what we showed is that on these low-calorie feeding protocols, patients were actually losing significant weight. You can see here they went below baseline, and we did not measure or detect any significant increases in weight until day 8. In our follow-up observational study of higher calorie feeding, starting around 17 or 1800 calories per day, we were able to shorten the length of stay by 6 days and document a weight gain as early as 4 days. So that is what this clinical research program has really been all about, is asking how can we improve outcomes in these patients through nutrition strategies in the hospital. And we were encouraged at the time we were starting this work because it seemed like from all of the retrospective studies that had come out that if you could speed up weight gain, you could improve outcomes. So several studies showed that patients who gained more weight in the hospital, either faster or more weight, did better at 1 year. Our psychiatry director, Daniel LaGrange, showed that in outpatient psychotherapy, weight gain of 1 to 1.5 pounds per week predicted clinical remission at the end of treatment. And another group by Angela Guarda showed that weight restoration would be quote unquote half price in terms of healthcare costs if you could just speed things up from 1 to 2 pound gains per week. So that was the preliminary work that formed the basis or motivated this original trial that we started in 2015. It was called the STRONG trial. And it's a multi-site trial. Our partners are Stanford. The Division of Adolescent Medicine there has a very similar eating disorder program as we do. And this work was funded by NICHD. And what we did is very straightforward clinical trial. We had 2 groups. The red group is the high calorie group. The blue group is the low calorie group. You can see we didn't go as low as 1200. We felt that was unethical given what we know about these kids languishing in the hospital on low calories. So we started at 1400. The low calorie group goes more slowly in terms of advancement. The high calorie group goes quickly, increasing by 200 calories every day. And here is our consort flow diagram. Just really quickly, you can see we had 60 in each group assigned to treatment. And in a modified intention to treat, we had 60 in the high group, 51 in the low calorie group. And just to show you briefly, this is the schedule of procedures. So we follow the kids every single day in the hospital, and then at 5 time points during the outpatient phase over 1 year. And we're looking at not just vital signs I mentioned, but also, of course, height and weight, electrolyte shifting, and then we have psychological measures as well. So remember I talked about electrolyte repletion. Here's the protocol that we developed, and really nice thing about having both sites, us and Stanford, with the same electronic medical record, is that we're able to embed this electrolyte repletion protocol into the medical record so that if one of our study patients does go low, the physician orders automatically populate. So as best we could, we had one protocol for the safety outcomes so that we could really, as best we could, measure the electrolyte shifting that would indicate risk for the refeeding syndrome. And here are the results. So cutting to the chase, what can you see here? You can see here that the graph looks very similar to the preliminary work. The high calorie group is shown in orange, and you can see that they gain weight from starting day 1 right out of the gate. The low calorie group, as we expected, they had significant weight loss on day 2 and had a real delay and less weight gain overall. The second figure is the survival analysis of the primary outcome. So this is the main outcome, main short-term outcome of the trial. And what you can see here, if you're an astute observer, you'll notice this is a bad graph, right? Because the colors are reversed. Here on the weight gain graph, blue is low calorie. Here, blue is high calorie. So keep that in mind. But what you can see here is for the reversal of those vital signs, those 5 vital sign instabilities that I showed you in the beginning, you can see that the high calorie group steps up to reversal more quickly. And so this was really our main efficacy outcome, to show that high calorie refeeding was more efficacious than low calorie. And this table breaks it down just a little bit more consumer-friendly way. The high-calorie group achieved medical stability 3 days sooner. Heart rate was restored 4 days sooner. Weight gain was 0.8 kilos greater. Length of stay was shortened by 4 days. And using national data sets, our health economist, who was Leslie Wilson, showed that the cost efficacy was almost $20,000 per participant. So in other words, this 4-day savings in hospital amounted to almost $20,000. So the question that you may be asking, if you're a physician especially, is to say, okay, well, that looks great. Things were moving faster, but what about the electrolyte shifting? Because the concern could be that you can feed patients more quickly, but that their electrolytes are bordering on, catastrophe, and that the physicians are running around trying to replete them. And actually, we really did not find that to be true. You can see here that for hypophosphatemia, which is considered the quote unquote hallmark feature or hallmark risk factor for the refeeding syndrome, only 4 kids in the high calorie group developed hypophosphatemia requiring electrolyte supplementation versus 3 kids in the low calorie group. So we really did not find electrolyte shifting to be unmanageable or higher in any respect in the high calorie group. The most common shifts were in magnesium. And in fact, they went in the opposite direction. More of the patients in the low calorie group developed hypomagnesemia. We did find, however, and this was unexpected, that the naders occurred earlier in the high calorie group. So this is actually something to really keep in mind, that now that we are feeding kids at higher calorie levels, we need to be keeping an eye earlier for their electrolyte shifting. This slide now shows the long-term outcomes. So this was a paper by the Stanford colleagues reporting then on what happens during the 12 months of follow-up. And what you can see is a little bit of a boring slide. There's just no difference in clinical remission. This is a dual definition of clinical remission. It includes weight restoration, and the other piece is cognitive recovery. And what they found is that there is no difference in the high versus the low calorie group in rates of clinical remission. So the upshot is that those savings that we showed in the hospital were maintained long-term. The other big question that was finally settled by my colleague Erin Accurso, shown here. She is one of the psychologists in our program, and she is an assistant professor on the psychiatry side. She measured mealtime distress, because one of the big concerns with high calorie refeeding was that, in fact, may have the unintended consequence of creating extreme psychological distress. And what she did show and publish is that actually there was no difference in mealtime distress when you compare high versus low calorie refeeding. So the high calorie refeeding itself did not predict more mealtime distress. And in fact, she showed some really nice associations with baseline characteristics. And no surprise, kids who enter the hospital with lower distress tolerance are the ones who had the highest mealtime distress. So all of those pieces combined, I'm really happy to say that these results have been rapidly translated into clinical guidelines. So we have in the US by the Society for Adolescent Health and Medicine, for example, in the UK, the Royal College of Psychiatrists, as well as the National Institute for Healthcare Excellence. That's nice. The new German S3 guidelines have incorporated high calorie refeeding, and then also just kind of the like physician references. Like if you look up refeeding anorexia in UpToDate or in Medscape, our data come up. So that's really exciting. And I feel like we've done a very good job translating these and not having our results kind of get stuck in our ivory tower, so to speak. But, won't linger too long on being proud, because turns out there's a lot of work to still be done. And as good as high calorie feeding may seem compared to the old standard of care, it turns out it's really not, it's not really great for everybody. So we're doing a new trial, and I'll tell you what motivated that. The new trial is the same group. It's us and Stanford, also funded by NICHD, very cleverly called, instead of just strong, it's iStrong, because it is individualized. And how is it individualized? So one of the big developments that occurred during the time we were proposing the old strong trial is that the diagnostic features for anorexia nervosa changed. So they changed right around the time we were proposing the study. This was back in 2013. Prior to 2013, in order to be diagnosed with anorexia nervosa, you had to have low weight. It was one of the diagnostic criteria, and it was less than 85% of expected. It wasn't very specific, but bottom line is that you had to be underweight in order to be diagnosed with anorexia. And so this was a growth curve that would be typical for our patients back in those days. A kid who was kind of growing along the median, fell off the curve, diagnosed with anorexia down here, and then gain weight during treatment as expected. Well, what happened at 2013 when the new diagnostic and statistical manual came out is they included a new category, and that new category is called atypical anorexia nervosa. Atypical anorexia nervosa describes patients who have all of the psychological features of anorexia nervosa, but they are not underweight. So here's an example of a growth curve of a kid who was growing along here on the higher end, and came in and here still, at what would be considered a normal weight, but extremely weight reduced at around the 25th percentile. And this patient was admitted to our service and diagnosed with atypical anorexia nervosa. So we luckily decided to include this diagnosis in the STRONG trial, even though we didn't really know what it would look like or how it would play out. At the time, you can imagine, because anorexia nervosa required low weight, all of the studies prior to 2013 associated low weight with more severe illness. So patients who had lower weights were more bradycardic, they were more hypotensive, they had lower bone mineral density, more hematologic abnormalities, higher LFTs, and they were deemed at greater risk for the refeeding syndrome. So this was really ingrained, and I have to say, I feel it's still ingrained in our thinking today, where we sometimes are very tempted to just say, oh, this patient is really skinny. Oh boy, they've lost a lot of weight. They're going to be really ill. Well, we looked among our patients, the 46 patients who we enrolled in our clinical trial with atypical anorexia nervosa, and we compared them at baseline to those with typical anorexia nervosa. And what we found is they are just as likely to have amenorrhea. They had a very similar percentage of body mass loss. Both groups had lost about 20% of their body mass. Almost all of them in both groups were bradycardic, and the next most common medical instability was orthostasis, you can see here. So I should go back one slide and just say there are many different ways to look at this, but the upshot is that the patients with atypical anorexia nervosa were just as ill as those with anorexia nervosa. And we published this finding, and there was one other group, Whitelaw and Sawyer, who published a similar study, and really helped to contribute to this acknowledgement of the severity of the malnutrition in atypical AN, that atypical AN wasn't somehow, anorexia light, that it really was a state of malnourishment and truly was an illness. Now, remember I hinted that while we were really excited about our improvements in refeeding protocols, well, turns out when you look at not just high versus low calorie refeeding, but you break it down by 4 groups to take into account diagnosis, the only group that really does well on high calories is the AN group. And you can see that shown in red. So it's clear patients with anorexia nervosa do well if they are refed or treated with high calories. The problem is for the patients with atypical AN, either in green or atypical AN in blue, it doesn't matter whether they were on low or high calories, they did poorly. They looked like they were on low calories. They straggled behind, and they took significantly more time to restore in hospital. And I'll just focus you in here. You can see here days to medical stability and days to restore heart rate, well, almost 2 days longer in the patients with atypical anorexia nervosa versus the kids with anorexia nervosa. They also gained less weight by every metric. They gained less kilos, they gained less percentage median BMI. The rate of weight gain was also slower. So what we really zeroed in on here with the kids with atypical anorexia nervosa is this idea of refeeding dose. If you take the number of calories that they received, whether they were on high or low treatment, and you divide it by their body weight, it was just simply too low. And here I can show that a little bit more easy to look at. This is calories per kilogram of body weight, and it's not rocket science. It simply just shows that patients with atypical AN, because they have a larger body habitus, they received a lower dose of calories. So I feel like for people who are physicians, this probably seems like a no-brainer because you probably often do this with medications, for example, where you dose per kilo of body weight. There may be other treatments where you take into account, like infusions, where you take into account kilograms of body weight. Some treatments might take into account, for example, surface area. But for nutrition, we really were kind of like in the Stone Ages, you know, with the high versus low calories, and we had not considered the body size of the individual. So when we did that, what we simply showed is that for patients who have atypical, the dose was inadequate. And that, in fact, is what explained the poorer outcomes, including longer time to stability, poorer weight gain, and longer length of stay in hospital. So the new trial is now focusing on this concept. And what we're doing is we're comparing a 50 calorie per kilogram dose to what is now the new standard of care, which is high calorie feeding. And this will just be, again, in some ways, very straightforward. We will look at efficacy, we'll look at safety, but we are doing some really exciting work around developing new benchmarks of recovery. And so we will be looking at metabolic, hormonal, and psychological outcomes that might be better than weight to indicate when patients with atypical AN have achieved recovery. So we're looking at reversal of that HPG axis suppression. So we'll be looking at return of menses. We'll be looking at sex hormones in boys and girls. We're using T3 as a marker of metabolic rate. And then we're looking at starvation stress, so to speak, with cortisol. We also are measuring metabolic rates. So we use a handheld indirect calorimeter. It's called a MedGem. And we perform that when they're admitted to hospital and when they leave. And then Erin Accurso, the colleague who I showed you who did that nice mealtime distress work, has added some very specific psychological measures that might be informative in kids who have a history of overweight. So our atypical kids might have some unique pathology related to perhaps stigma due to their weight prior to illness. So I'm most excited about this third aim, and stay tuned. Okay, so I've told you about our old trial, the STRONG trial, and how that really provided the evidence for this new standard of care, the high calorie feeding, but that we have a growing number of kids who have atypical anorexia nervosa. They make up about a third to a half of our inpatient service now, and they don't do well on high calorie feeding. So that's the new trial, the individualized STRONG trial. And then I just thought I would end by talking about a few exciting directions that we're going with the work. One of them is looking at the gut microbiome. You may know that patients with anorexia nervosa have marked dysbiosis. So it's no surprise, right, that starvation really, the gut microbiota really take a hit during starvation. But the idea that was really developed early on in studies of children in developing nations is that the dysbiosis contributes to this self-reinforcing pathogenic cascade of malnutrition, so that the gut becomes malnourished and it cannot, for example, extract nutrients, and therefore it perpetuates the state of malnutrition. I jumped ahead to that last piece, this idea of poor nutrient extraction, and that has been shown actually. Von Schwarzenberg, in collaboration with the Turnbaugh Lab, published a really nice study showing that microbial transplants from weight-reduced human patients confer resistance to weight gain and high-fat diet in mice. So that is seen, this idea of weight gain resistance. It looks great for patients who potentially are trying to avoid weight regain after successful intervention. But for our patients, that's a disaster. It's something that we're struggling with every day, is this, at least is evidenced clinically, that it's very, very difficult for them to gain weight, even on high-calorie diets. So we're sending some stool samples over to the Turnbaugh lab for them to burn in their bomb calorimeter, to see if we can just look at how much energy is left over in the stool, and maybe this malnourished gut is not efficiently extracting energy from stool. The other area that we'll look at really carefully is short chain fatty acid production. There is mounting evidence, or at least one consistent finding over the handful of studies that have been done in patients with anorexia nervosa, is that they have lower production of short chain fatty acids. And we're going to look at whether, or one hypothesis is that potentially that could contribute to the hypometabolic state. You may know that gut microbiome produces the short chain fatty acids that are produced by the gut fulfill up to 10% of human energy requirements. So just a reduction there might contribute to hypometabolism. And then, of course, the short chain fatty acids are also really important modulators of appetite regulation, for example, PYY and GLP-1. And so I've paired up with this work. We were lucky to get a microbiome pilot study, the BCMM pilot study. I paired up with a junior faculty person, Ryan Rampersad, who some of you might know from psychiatry. He's an MD PhD who has training in basic molecular biology, and we'll be looking at some of those concepts that I just mentioned in relation to illness severity and response to refeeding. The other new direction that I just wanted to mention, we're very, very excited about, is body composition. So along with this new focus, or I should say this intensified focus on malnutrition, you may be aware that we finally now have new guidelines for detecting and diagnosing malnutrition. And the global criteria now include body composition. So yes, weight is still in there, but you can see it's become a small piece of the puzzle. For adults, BMI less than 20 is one criteria, but now body composition is really featured alongside functional criteria like reduced strength and etiologic criteria like reduced food intake. And so we have always wanted to look at body composition, but we just could not get our kids out of the unit while they're medically unstable and over to DEXA or MRI. So we've been in collaboration with John Shepard's group. Some of you may remember Roberta. I'm sure you remember John Shepard from when he was here at UCSF. He's at University of Hawaii now, but I've been collaborating with him for over 10 years. And his group has really developed and validated this three-dimensional optical body imaging. Many, many studies now showing that it has excellent accuracy compared to DEXA in children, in adolescents, in adults, across ethnicities, in relation to strength, in relation to biomarkers, and also to pick up changes in body composition during intervention. So we used 3D optical imaging to show that we could accurately detect malnutrition in a sample of 95 participants from John Shepard and Steve Hemsfeld's Shape Up trials. We also showed that we could detect losses of lean mass during the weight loss phase, and that we could detect gains of fat mass during the weight regain phase. And so we are really excited to move ahead. One of my colleagues, Amanda Downey, who is a somewhat newer faculty in our division, has gotten a NORC pilot grant, and she is using 3D optical imaging to look at the efficacy of refeeding and markers of malnutrition in our hospitalized patients. Here's our research team. They're such good sports. They're willing to come to work in their workout outfits and get scanned. This is what the 3D scanner looks like. It's small, it's portable, and we're starting the pilot study now to do those body scans. The other area that the 3D optical imaging really has potential is in providing insight into this hypometabolic state. And so what Amanda has been focusing on part is this idea of the energy deficit that brings our patients into the hospital. So we know that if your body size is normal for your own body habitus, you have an expected metabolic rate that matches it. And all things being equal, if you are in energy balance, your metabolic rate and your tissue mass should match, and you should stay in energy balance. Of course, our patients have created an energy deficit either by restricting or by overexercising, and usually both. And what Amanda has showed is that this deficit is on average 840 calories per day in our patients. So they're in the red, so to speak, by over 800 calories per day. And what the body composition can start to discern is the contribution to this hypometabolic state of just having a smaller body. So as you can imagine, as people lose weight, smaller body burns fewer calories. And you can look at that with body composition because, of course, lean mass is much more metabolically active than fat mass. But there's also something that's not explained by body composition, which is this idea of adaptive thermogenesis or energy efficiency at the cellular level, which may be due to low leptin and a number of other things. So we're hoping to look at this and start to get a feel for the relative contribution of adaptive thermogenesis versus frank tissue reduction in creating the hypometabolic effect or hypometabolic state in our patients. And one thing that Amanda has just shown in preliminary data is that kids who have a larger energy deficit, no matter what their body size is, require more time in hospital to restore medically. So we'll be, just to finish up here, we'll be creating a 5-compartment. Our goal is to create a 5-compartment model of body composition. This has been done in patients with severe malnutrition due to anorexia nervosa. And what the study showed, this is a study by Comiskey et al., they showed that there is about an 18% reduction in bone mass, almost 70% reduction in adipose tissue, 21% reduction in skeletal muscle mass. And what we're particularly interested in is this 25% reduction in residual mass. And the reason we're so interested in this is because this compartment, this residual compartment, contains the highly metabolically active organs like the heart, the kidney, of course, the brain. But specifically, the heart, the kidney, and the liver have a big role in the medical instabilities that we're interested in hospitals. So electrolyte stability, fluid stability, and then autonomic regulation. So that's what we hope to do with that body scanner, is look at all of these compartments, but really trying to focus in on the role of residual mass reduction and refeeding outcomes. So I just want to end there by thanking our group. These are all the people who are working on iStrong from Stanford, from UCSF. We had Barbara Mashiski, who I was talking about with Roberta, from UCLA. The staff who's really been supporting the work, and we also have an international data safety monitoring board shown here. I also wanted to recognize my collaborators at University of Hawaii. Here's John Shepard, who some of you may remember, and the other PI on the Shape Up studies is Steve Hemsfeld from LSU Pennington. And so I'll end with that, and of course, happy to take any questions.
you, Andrea. That's great. Congratulations on getting your work into the guidelines. That's a
Thank
fantastic
you.
accomplishment. Well, we will open it up to questions. I had a couple of kind of thoughts, questions. One was around boys. I think you said it. You had 5 boys in the strong study. Wondering if there's any differences now with when you're focusing on the atypical anorexia nervosa and how
Yeah.
that, may inform your future directions.
It's a really important question. If I might have misspoken, I meant to say we have 5 boys in the current study, in the
Oh,
ICE
okay, okay.
study. And atypical appears to be more common or slightly more common in boys, but I think it's just by nature of their body size. They tend to have, because they have larger body size. So we only have 5 boys. But we finally now we said we can't keep excluding boys or we'll never understand. So we did fight to include boys. We only have 5. And we will be looking at their testosterone and then how that improves over time as they improve nutritional status. But I don't know yet. Who has really done some great breakthrough work on boys with eating disorders is Jason Nagata, my colleague and one of my mentees, who I'm very proud of. But we have a medical registry for our whole program. And what he has shown in a couple of publications now is that boys require, again, no surprise, more calories in the hospital and more time to recover. So it may be very similar to our atypical kids where we just were not really paying attention to their larger body habitus. They need more nutrition to kind of turn that catabolic train around.
So my other question, please, anyone raise your hand or pop in if you like. My other question was around the psychological data. I was actually, I was surprised that you didn't see any differences there. And I was wondering how sort of this framing around nutrition may influence that, or if you have, malnutrition, I guess, malnutrition, how that may influence that.
Yeah. It is interesting that we don't see any differences. And my hypothesis, and I really don't have a glimpse, so I can't say now in the new study whether this will be true. But I think that our tools are just too rudimentary. They were designed for kids with typical anorexia nervosa, which meant underweight girls. And I hope that what we find is that we just haven't paid the appropriate respect and the appropriate credence to kids who have a range of cognitive cognitive and behavioral changes around their eating disorder that's could be related to their gender, to their sex, or to their body size. I think we just don't know. And we keep using these old tools. We're like, oh, there's no difference. Oh, there's no difference. But I'm not convinced yet that that means it's the same or that there's no effect.
Mel, you want to go ahead?
Sure. Thanks, Andrea. Wonderful talk and wonderful research you're doing.
Thanks, Mel.
I'm curious. Steve Heimsfeld's done some work with doubly labeled water, and I'm curious whether you've looked at the total energy expenditure. The thought being, I guess, more for when they're recovering out after discharge. Before, I know there's a study. I'm trying to remember when, 30 years ago, looking at doubly labeled water and anorexic and suggesting that their total energy expenditure is very high before they get admitted because of the increased exercise that a lot of these girls are doing. Anyway, just
Yeah,
thought.
Steve, I would love to do doubly labeled water. And we did just kind of like a back of the envelope, a little quick sketch out to see if we could afford it. And if we considered like all the different time points that we were trying to capture, it was something like $17,000 per participant. I mean, it was crazy. So we just haven't been able to afford to do it. But we did talk to Kathy Mulligan, who I know that you know, and she said she had like some kind of isotope left over that she might be able to lend us. But it's a great idea, and it just hasn't been feasible for us yet. And we're using these my research assistants know really well. It's not a good tool, and there's a lot of problems with calibrating it. So I think I think we would be so great if we could move to doubly labeled water.
So Naomi put a comment in. She had to hop off for clinic, but she said, thank you for the important work and great presentation.
Oh, great.
Is there anyone else who'd like to unmute and ask a question? I see we do have a good GI complement here. So we had a couple endocrinologists, but it looks like they had to get off as well. So. Well, thank you so much for the great presentation, and everyone, please join us again in March.
Yeah, thank you for having me.
All right, take care. Bye.